Okay, let's dive right into this. For decades, the whole idea of bringing back the Tasmanian tiger, the Thylacine, it's felt like pure science fiction.
Completely. But that's just not the world we're in anymore.
No, the paradigm is totally shifted. We are not talking about a theory here.
We are talking about a very serious, very well-funded scientific effort. As of late 2025, this is active genetic engineering.
So who are the main players? Who's leading this charge?
Well, the heavy hitters are Colossal Biosciences, a US biotech firm. You probably know them from their work on the woolly mammoth. And they're collaborating with the University of Melbourne's TIGRR lab, which is led by Professor Andrew Pask.
And the fundamental problem they're facing seems almost impossible. How do you bring back an animal when the last one died almost 100 years ago? You don't have a living cell to start from.
That's the key. And that's why they are emphatically not doing traditional cloning. This isn't a Dolly the sheep situation.
Because that would need an existing nucleus from a living cell.
Exactly. This relies entirely on gene editing, specifically CRISPR, to perform what is essentially a biological reconstruction.
So let's talk about the ingredients. The first piece is the blueprint, the DNA,
which they managed to extract from a thylacine specimen that was preserved in alcohol for over a century.
Which is just staggering in itself. And the second piece?
The second piece is the living template, the surrogate, and for that they're using the fat-tailed dunnart.
A tiny mouse-sized marsupial.
It is, but it's the thylacine's closest living relative. It provides the sort of basic genetic scaffolding they need to do the work.
So the technique isn't copying, it's more like a molecular conversion. They're editing the dunnart's genome.
That's right. Swapping dunnart genes for thylacine genes bit by bit.
And the goal is to transform a dunnart cell into basically a thylacine cell,
which can then be grown into an embryo using stem cell tech and artificial reproductive techniques.
And the progress they've made here, it's moved from just difficult to genuinely astounding.
They've successfully reconstructed a thylacine genome that is 99.9% complete.
99.9%?
To put that in perspective for you, this is the most complete ancient genome of any species ever sequenced. They have the instruction manual.
But they went even further than the DNA blueprint. There's this world-first achievement with RNA.
Yes, this was a massive leap. They isolated RNA from preserved soft tissue, the tongue, the nose, the brain.
I thought RNA degraded almost instantly. How big of a hurdle did they just clear with that?
It's transformative. See, DNA tells you what the animal could be. RNA tells you which of those genes were actually switched on when it was alive.
So it tells you about the biology in action?
Exactly. Things like its sense of smell, specific taste receptors, its nocturnal vision. It's not just the blueprint, it's the user's guide.
And that let them prioritise their work. I saw they have already made over 300 specific genetic edits to a dunnart cell to make it more thylacine-like.
And those edits are precision strikes. They're targeting traits linked to those unique marsupial carnivore features.
So, given all that complexity, what's the timeline? When might we actually see something?
Well, the optimistic estimates suggest we could see a thylacine joey within the next 6 to 10 years.
6 to 10 years, wow.
Though rewilding, of course, that would take much, much longer.
But here's the catch, and this is the resulting animal will not be a 100% copy of the extinct thylacine. It's going to be a hybrid with some dunnart DNA, filling in those tiny gaps.
And that leads directly to the primary criticism of this whole thing.
Which is?
Why pour all these resources into bringing a species back when you could be using that money and expertise to save currently endangered species, like the Tasmanian devil?
It's a powerful ethical question, but the counter-argument is the tech spillover.
Precisely. The specialised tools being built for this, like the artificial marsupial womb, are already helping current conservation efforts.
So developing that womb accelerates research into diseases that affect living marsupials, like the devil facial tumour disease. It gives researchers a controlled environment to study early development.
So the innovation helps species right now.
So we're looking at a future where a creature that looks and acts like a thylacine is reborn using science that's also helping save living animals.
That's the hope. But it leaves us with a really provocative thought. If the first animal we resurrect is, by definition, a hybrid, a genetic mosaic, what does that do to the whole concept of a species in the future? What new conservation opportunities or, what new challenges does creating this kind of mosaic present to you?